Assalamu alaikum. Today we want to talk about the citric acid cycle. Now, generally speaking, organisms they get far more energy from nutrients by aerobic oxidation than by anaerobic oxidation. In chapter 17, we talked about glycolysis, where one molecule of glucose was converted to two molecules of pyruvate. Now, in this chapter and the next one, we will see how 30 to 32 molecules of ATP can be produced from each molecule of glucose in complete aerobic oxidation to carbon dioxide and water. There are three processes that play key roles in aerobic metabolism: citric acid cycle, the one we are going to talk about in this chapter, electron transport, and oxidative phosphorylation. These two topics we will talk about in chapter 20. Now, metabolism consists of catabolism, which is the breakdown of large molecules into smaller ones. This is oxidative in nature. Energy yielding, and anabolism, which is reductive in nature and energy requiring. Now, the citric acid cycle is referred to as being amphibolic, meaning that it plays a role in both catabolism and anabolism. Now, this figure shows us the central relationship of the citric acid cycle to catabolism. Here, carbohydrate metabolism goes into three stages. Stage 1, acetyl-CoA production. We will see this. Then, stage 2, acetyl-CoA oxidation in the citric acid cycle, as we will talk about. And then stage 3, electron transport and oxidative phosphorylation. One important note to remember is that all metabolic pathways operate at the same time, even though we talk about them separately. Now, the citric acid cycle is referred to as being amphibolic, as I talked about. It has two other names. One is the Krebs cycle, in honor of Sir Hans Krebs, who first investigated the pathway, and he received a Nobel Prize in physiology or medicine in 1953. The other name is the tricarboxylic acid cycle or TCA cycle, since some molecules involved in the cycle are acids with the three carboxyl groups. Let's begin with the overall pathway of the citric acid cycle. In eukaryotes, glycolysis occurs in the cytosol, whereas the citric acid cycle takes place in the mitochondria. Now, most of the enzymes of the citric acid cycle are present in the mitochondrial matrix. Now, the reactions of the cycle take place in the matrix, except for one in which the intermediate electron acceptor is fed. The enzyme that catalyzes the FAD-linked reaction is an integral part of the inner mitochondrial membrane and is linked directly to the electron transport chain, as we will see later. Now, this figure shows us the structure of a mitochondrion, where you can see here the inner membrane, outer membrane, the matrix, and the cristae. Now, the reaction of the cycle, as I said, takes place in the matrix, except for one in which the intermediate electron acceptor is fed. Now, overview of reactions in the citric acid cycle. Under aerobic conditions, pyruvate produced by glycolysis is oxidized further, carbon dioxide and water as the final as water and carbon dioxide are the products of the oxidation. Now, at the beginning, pyruvate is oxidized to one carbon dioxide molecule and to one acetyl group, and then this becomes linked to an intermediate coenzyme A. Now then the acetyl-CoA enters into the cycle where two more molecules of carbon dioxide are produced for each molecule of acetyl-CoA that enters into the cycle, and electrons are transferred. Now, immediate electron acceptors in the cycle are NAD+, this oxidizing agent, NAD+ reduced to NADH, and FAD takes up 2 electrons and 2 hydrogen ions to produce FADH2. And we will see that the final electron acceptor is oxygen. So electrons are passed from the reducing equivalents in the form of NADH and FADH2 through several stages of an electron transport chain with a different redox reaction at each step. Now the final electron acceptor is oxygen with water as the product. Now the cycle also produces GTP, a high energy compound, directly, and this is equivalent to ATP. And then the cycle is completed by regeneration of oxaloacetate from succinate steps. Now the citric acid cycle has 8 steps, as you can see in this figure, from 1 to 8, and these are catalyzed by different enzymes. 4 of the 8 steps, namely 3, 4, 6, and 8, are oxidation reactions. Now the usual oxidizing agent is NAD+, in all except the one in step 6 here, where in step 6 the enzyme succinate dehydrogenase and the oxidizing agent is FAD. And we notice here in step 5 the one molecule of GTP is produced upon phosphorylation of GDP. And as I said, this is equivalent to the production of 1 ATP. Now let's begin with a reaction prior to the cycle. How pyruvate is converted to acetyl. Now pyruvate, which can come from several sources including glycolysis, moves from the cytosol into the mitochondrion via a specific transporter where an enzyme called pyruvate dehydrogenase complex is responsible for its conversion to carbon dioxide and acetyl production of acetyl-CoA. Now, this enzyme consists of, or this enzyme complex consists of up to 5 enzymes, and these are pyruvate dehydrogenase, dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase, pyruvate kinase, and dehydrogenase phosphatase. Now the first 3 are involved in pyruvate to acetyl-CoA, as we will see in the mechanism. The other 2 are used to control the pyruvate dehydrogenase. Now this enzyme requires 5 coenzymes. Coenzyme A, TPP stands for thiamine pyrophosphate, FAD, lipoic acid, in addition to NAD+, and a metal ion cofactor Mg+2. Now, pyruvate first loses CO2. If we look at the mechanism here, and hydroxyethyl TPP is formed, then active form of lipoic acid is bound to the enzyme. Dihydrolipoyl transacetylase by an imide bond to the epsilon amino group of lysine. Then this hydroxyethyl group is oxidized and transferred to a sulfur atom of the reduced form of lipomide, which is then reduced to dihydrolipomide. Acetyl group is transferred to the sulfhydryl A, and then this dihydro is oxidized to lipomide. If we look at the mechanism here, you see we have here step 1 here. You see the decarboxylation of pyruvate occurs through the formation of hydroxy TPP, then transfer of the 2 carbon unit to lipoic acid. In step 2, followed by formation of acetyl-CoA here, and then lipoic acid is reoxidized in this last step. Now we want to talk about why pyruvate dehydrogenase has a compact arrangement. It is a multi-enzyme complex. It has stages over an arrangement in which various components are more widely dispersed. Now, the various stages of the reaction can take place more efficiently. Why? Because the reactants and the enzymes are close to each other, proximity to each other. And the second advantage of a multi-enzyme complex is that regulatory controls can be applied more efficiently in such a system than in a single enzyme molecule. We'll talk about the control in later sections. Now let's go and describe the individual reactions of the citric acid cycle, beginning with this table. This table shows us the 8 steps or 8 reactions with 8 different enzymes. Going back to step 1, formation of citrate. Acetyl-CoA reacts with oxaloacetate to form citrate and coenzyme A. As you can see here, the enzyme is citrate synthase. It's also called the condensing enzyme because this reaction is a condensation due to the formation of a new carbon-carbon bond. Delta G naught prime for this reaction is negative, it's exergonic, since the hydrolysis of thioester releases energy. Thioesters usually are considered high energy compounds. Now step number 2, isomerization of citrate to isocitrate. Now citrate is achiral and it's converted to chiral isocitrate in the presence of enzyme called aconitase, and it requires iron plus 2. Notice here, isocitrate has 2 chiral carbons here with the possibility of 4 isomers, but only one of them is being formed in the cycle. They found that fluorocitrate is a potent inhibitor of aconitase. Now, if we look at this figure to see which shows us the 3-point attachment to the enzyme aconitase makes the 2 groups CH2COO and CH2COO stereochemically non-equivalent. Actually, what happens is that the enzyme forms an unsymmetrical 3-point attachment to the citrate molecule, as is shown here in this figure. Now, the reaction proceeds by removal of a water molecule from the citrate to produce cis-aconitate, and then water is added back to the cis-aconitate to give isocitrate. Now this intermediate remains bound to the enzyme during the course of the reaction, and there is some evidence that citrate is complex to the iron 2 in the active site of the enzyme in a circular conformation. Now step 3, formation of alpha-ketoglutarate and CO2. This is known as the first oxidation in the cycle, and because it's accompanied by CO2 removal, it's called oxidative decarboxylation. The reaction is catalyzed by an enzyme called isocitrate dehydrogenase. Notice that the reaction occurs in 2 steps. This is step 1 and this is step 2. First, isocitrate is oxidized to oxalosuccinate, which remains bound to the enzyme, and then oxalosuccinate is decarboxylated and the carbon dioxide and alpha-ketoglutarate are released. This is the first reaction which produces a reducing equivalent in the form of NADH, which later will produce ATP, as we will see. The enzyme is an allosteric enzyme. We will talk about its control later on. Now, step number 4, formation of succinyl-CoA and CO2. This is known as the second oxidative decarboxylation of alpha-ketoglutarate to form succinyl-CoA. It is the second oxidation in the cycle, of course. The enzyme is a multi-enzyme complex that requires the same 5 coenzymes and metal cofactor as were used in pyruvate dehydrogenase. As you can see here, both dehydrogenases are similar with this respect in their requirement. The reaction takes place in several steps, and again there is the requirement for these coenzymes. The reaction is highly exergonic, as is the one catalyzed by pyruvate dehydrogenase. So at this point we can say that 2 molecules of CO2 have been produced by the oxidative decarboxylation of the citric acid cycle. Removal of the CO2 makes the citric acid cycle irreversible in vivo, but not in vitro, where each separate reaction is reversible. Then we go to step 5. Step 5, formation of succinate. Now in this step of the cycle, a thioester bond of succinyl-CoA is hydrolyzed to produce succinate and coenzyme A, and accompanying reaction is the phosphorylation of GDP plus PI to form a GTP. Now the whole reaction is catalyzed by the enzyme called succinyl-CoA synthetase. Now synthetase is an enzyme that creates a new covalent bond and requires the direct input of energy from a high energy phosphate. Now with the phosphate group, this one is covalently bonded to the enzyme and is directly transferred to GTP. The phosphorylation is endergonic reaction and it gets the required energy from the hydrolysis of this thioester bond. And this is called coupling, and coupling is what metabolism is all about. Now, in the next 3 steps in the TCA cycle from 6 to 8, succinate 4-carbon is converted to oxaloacetate to complete the cycle. By the way, the production of GTP, which is equivalent to ATP directly from the cycle, is referred to as substrate level phosphorylation to distinguish it from oxidative phosphorylation, which we will talk about later on. So now, step number 6, formation of fumarate and FAD-linked oxidation. Succinate is oxidized to fumarate that is catalyzed by the enzyme called succinate dehydrogenase. This enzyme is an integral protein of the inner mitochondrial membrane. whereas the other individual enzymes of the cycle are in the mitochondrial matrix. Here you see Fad is the oxidizing agent, and the electron acceptor, rather than an Ad+, is also covalently bonded to the enzyme succinate dehydrogenase and is called a flavoprotein because of the presence of FAD with its flavin moiety. It's also called a non-heme iron protein or an iron sulfur protein. Succinate dehydrogenase contains iron atoms but does not contain a heme. That's why it's called a non-heme iron protein. Now the later name, iron sulfur protein, refers to the fact that the protein contains several crystals that consist of 4 atoms each of iron and sulfur. Again, FADH2, this reducing equivalent, can be later give ATP, as we will see in the next chapter. Now, step number 7, however, formation of L-malate is catalyzed by an enzyme called fumarase. This is a hydration reaction, addition of the water across this double bond to give us L-malate. Malate has 2 enantiomers, L and D malate, but only L-malate is produced in this reaction. Then the final step in the cycle, step 8, regeneration of oxaloacetate. This is also the 4th and final oxidation step. Malate is oxidized to oxaloacetate, and another molecule of NAD+ is reduced to NADH, as you can see here. The reaction is catalyzed by the enzyme called malate dehydrogenase. The oxaloacetate can then react with another molecule of acetyl-CoA to start another round of the cycle. Now, we can say that the oxidation of pyruvate by the pyruvate dehydrogenase complex here, which is prior to the cycle, and the citric acid cycle results in the production of 1 plus 2, 3 molecules of CO2. As a result of this oxidation, 1 molecule of GDP is phosphorylated to GTP. 1 molecule of FAD is reduced to FADH2, and 4 molecules of NAD+ are reduced to NADH. Of the 4 molecules of NADH produced, 3 come from the citric acid cycle, and 1 comes from the reaction prior to the cycle. Now, the overall stoichiometry of the oxidation reaction is the sum of these two, the pyruvate dehydrogenase complex reaction and the citric acid cycle reactions, and the overall reaction is summarized here. So, 1 pyruvate converted all the way to 3 CO2 or oxidized, with the result of 4 NADH, 1 FADH2, and 1 GTP. This is the one which I refer to as substrate level phosphorylation. Now these later, as we will see, eventual ATP production per pyruvate, the 4 NADH will produce 10 ATP, 2.5 ATP for each NADH, oxidized in the electron transport chain. 1 FAD produces 1.5 ATP for each FADH2 when it is oxidized in the electron transport chain, and the 1 GTP is equivalent to 1 ATP. So what we can say that these important reactions play a large role in the cycle contribution to our metabolism. One purpose of the cycle is to produce energy. It does this by producing GTP directly and by producing reduced electron carriers in the form of NADH and FADH2. This is FADH2. The 3 decarboxylations mean that for every 3 carbons entering as pyruvate, 3 carbons are effectively lost during the cycle, a fact that has many implications to our metabolism, as we will see later in this chapter. End of part 1.